So the first speaker would be myself, Ryosuke, and I am a postdoc at the Technical University of Munich. Okay, this one, thank you. Working on neuroscience, originally from Japan. Today, I will very briefly talk to you about my work on landmark anchoring in zebrafish compass neurons, or how babyfish stay oriented. So imagine you're taking a short walk from a hotel in a new city. Even if you are unfamiliar with the environment, usually you can circle back to the hotel without even thinking. The reason why you have such sense of direction is because you have neurons in your brain that works like a compass, which fires only when you're facing in particular direction in the environment. Such compass-like neurons were first described in rats more than 3 decades ago, which is thought to sort of keep track of the directions by remembering the turns animals make. But for technical reasons, it has remained challenging to actually figure out how the connections among these neurons produce these non-trivial compass-like activities. So our research group decided to tackle this question in larval zebrafish instead of rodents. So these babyfish have a brain that shares the general sort of architecture with our own brains, but with a vastly smaller size, with 1,000 times less neurons compared to rats. So this small size makes it easy for us to understand mechanistically how the connectivity between these neurons produce interesting computational properties. So in this babyfish brain, we found a nicely arranged array of compass neurons whose population activity rotates clockwise and counterclockwise as fish makes turns left and right, just like a real compass. So in addition, using an electron microscopy-based circuit analysis, we found that these neurons form recurrent loop-like connections, which had been sort of predicted from theoretical studies. So far so good, but there's a big missing piece in this story, which is visual landmarks. So we humans heavily rely on visual landmarks to orient ourselves. For example, if I were in Munich and if I see the Alps, I can immediately decide I'm facing south. But this sort of associations between sort of visual landmarks and your orientation is entirely dependent on the specific environment you're in. So this is something your brain, or rather the compass neurons, need to learn. So I set out to ask if the fish compass neurons can learn to take advantage of these kind of visual landmarks, and if so, how? So to do so, I built an immersive virtual reality setup that's composed of a small projector and multiple small mirrors. While I look into their sort of brain using two-photon calcium imaging, the fish experienced the virtual cylindrical scenery, which rotated about them as fish made turns. Here, the activity of these compass neurons are shown as a blue heat map, where it's sorted by their preferred directions in y-axis and over time in x-axis. So you can see that this yellow line, which indicates the sort of basically direction that these brain compass were pointing as a population, nicely followed the orientation of these visual scene, which is plotted in black. In addition, if I look at the map, how the sort of preferred orientations of these compass neurons map onto the anatomical locations of these cells, I can see that they are nicely arranged in a circular fashion in each fish, as you can see from these nice contiguous rainbow patterns. But also there are some unique offsets among these individual fish. So this lack of stereotypy indicates that each fish learns to sort of map the visual landmarks onto their brain compass in different ways. Finally, we wondered how these compass neurons receive these kind of visual informations. And typically, in rodents, it's assumed that this comes from a visual cortex, but that's something this babyfish doesn't really have. Instead, we decided to focus on a nucleus called habenula, which is in purple here. So habenula is a highly conserved nucleus that's from jawless fish to birds and mammals. And they send these like long, dense axons to where the dendrites of these compass neurons are. So I decided to ask if this habenula is really required for the sort of landmark tracking by compass neurons by irradiating a strong laser and cutting these long axons. And before the cut, again, the direction of this like brain compass nicely followed in yellow the scene orientation, which is in black. However, after the cut of habenula axons, these two were no longer aligned. even though the brain compass was still pointing somewhere. So in summary, I showed you that Larval zebrafish brain has compass-like neurons, just like what's presumably supporting our own sense of direction. And using a VR system, I've shown you that these compass neurons can learn to follow landmarks. And finally, the new highly conserved nucleus called habenula is necessary for their landmark information, implicating it as an evolutionarily older visual pathway for navigation alternative to the visual cortex. And if you're interested in more details, the preprint is out a couple of weeks ago, so you can check it out. And thank you for your attention.